agriculture industry of the world has been facing serious problems due to microbial disease. The present study was carried out to know the gut microflora of two commercially important crops, P. pelagicus and P. sanguinolentus, for a period of 1 year from December 2008 to November 2009. Portunus, pelagicus, linus, and P. sanguinolentus herbs are two commercially important crop species that are widely harvested and consumed due to their excellent taste and nutritional value. Microbial poses a significant threat to the safety and quality of seafood products, including crabs. Pathogenic microorganisms such as bacteria, viruses, and parasites can lead to various foodborne illnesses upon consumption, causing adverse health effects in humans. These contaminants can originate from various sources including water, handling processes, and environmental conditions during the stage of harvesting, processing, transportation, and storage. This research aims to provide valuable insight into the type, levels, and source of microbial contamination in the crops, ultimately contributing to effective food safety measures and the sustainable management of crop fisheries. Next, let's proceed to the goal of study. So what is the goal of the study? The goal of the study was to comprehensively investigate and analyze the extent of microbial contamination in commercially important crops, Portunus, pelagicus, linus, and P. sanguinolentus herbs. The study aimed to identify prevalent microorganisms, understand the source of contamination, assess potential health risks associated with consumption, and provide insight to enhance food safety measures in the crab seafood industry. Next, we'll proceed to how was the study was implemented. The study was implemented through a multi-stage approach. First, sample of Portunus, pelagicus, and P. sanguinolentus were collected from various harvesting location and processing centers. The samples were subjected to microbiological analysis to determine the presence and the level of various microorganisms, including bacteria, viruses, and parasites. Additional samples of water, handling equipment, and processing surface were also collected to assess potential source of contamination. Microbial identification techniques such as PCR and cultural-based methods were employed to identify specific microorganisms. And also, statistical analysis was conducted to correlate contamination level with location, season, and processing practices. So now, let's proceed to the major findings. So, the major findings, there are studies revealed several significant findings. First, both Portunus, pelagicus, and P. sanguinolentus showed varying degrees of microbial contamination, with bacteria being predominant contaminants. And Vibrio species were identified as prevalent contaminants, with certain strains showing pathogenic potential. And also, contamination levels were influenced by factors such as geographical locations, handling practices, and processing methods. And lastly, water quality and cleanliness of processing equipment emerged as critical factors affecting contaminations. And now let's proceed to the importance of the study. This study actually holds immense importance for multiple reasons. It provides valuable insights into the specific microbial contamination risks associated with Portunus, pelagicus, and P. sanguinolentus, contributing to tailored food safety strategies. By identifying prevalent contaminants and their sources, the study aids in developing targeted mitigations and measures to reduce health risks to consumers. With that, the finding assists regulatory bodies, seafood processors, and harvest in implementing improved hygiene and processing practices to enhance crab product safety. So with that, along the similar lines, a parallel experiment could focus on the effectiveness of various processing and preservation methods in reducing microbial contamination in crab products. For instance, the study could compare the microbial loads in crabs subjected to different treatment methods such as high pressure processing, UVC treatment, and modified atmosphere packaging. And so, the experiment could also involve inoculating crabs with known contaminants and evaluating the effectiveness each method in reducing microbial population. So this research will provide practical insight into innovative preservation technique that could be implemented and enhance the safety and the shelf life of crab products in the market. I think that's all. Again, I am Lucila Caligaran, PSFR 3A. Thank you for watching.